Absolute pleasure to extend a hearty welcome to each one of you present here at the online refreshers course on Bioscience, Bioengineering and Nanotechnology organized by UGC-Malaviya Mission Teacher Training Center, University of Calcutta in Maulana Azad College, Kolkata. We are thrilled to witness such enthusiastic participants and a highly learned audience gathered to deepen their understanding of this significant topic through this refresher course. Today's seminar on bioscience, bioengineering, and nanotechnology aims to bring to light various aspects associated with this topic, thereby sparking thoughtful discussion and stimulating new ideas. In essence, these three fields form a powerful synergy. Bioscience provides the living blueprints, bioengineering provides the design and application framework, and nanotechnology provides the tool and material at the most fundamental scale to interact with and manipulate biological system effectively. We are fortunate to have with us today a distinguished speaker who is a noted figure in the field of medical research. I am introducing Dr. Sulogna Basu, who is scientist G at ICMR National Institute of Research in Bacterial Infections, Kolkata, formerly ICMR NICET. She leads active research in antibiotic resistance in bacteria causing neonatal sepsis. Her laboratory has been able to drive advancement in the fight against antibiotic resistance and neonatal healthcare. Dr. Basu's work has yielded numerous publications in the area of neonatal health and AMR. She is a fellow of both the National Academy of Sciences, India, and West Bengal Academy of Science and Technology. She is also as a member of the WHO Technical Advisory Group on Vaccine and Antimicrobial Resistance and member of the close group of AMR experts that supported the development of the first WHO global AMR research agenda in the human health sector. Dr. Basu's commitment to advancing scientific knowledge is reflected in her roles as an associate editor for two esteemed journals, Journals of Antimicrobial Chemotherapy, Antimicrobial Resistance, and Frontiers of Microbiology. Her laboratory also played a pivotal role in the multi-country Burners project funded by the Gates Foundation dedicated to advancing neonatal healthcare. The duration of this class is 80 minutes, and after completion of Dr. Basu's presentation, there will be an interactive session for 10 minutes to clarify your doubts and queries. As we set out on this intellectual journey, we hope that the session will offer all attendees a plethora of insights, enable fruitful interaction, and inspire fresh perspective about the topic at hand. Without further ado, let's begin our journey into the intriguing world of bioscience, bioengineering, and nanotechnology. I am honored to invite Dr. Basu, our distinguished speaker of the day, to share her valuable insights. Ma'am, kindly start your lecture.
Good morning, everybody, and thank you for the kind words. I would first like to thank the organizers for giving me this opportunity to come and share and talk to you about our research. So, I will talk less about the research, but I will primarily take you through how there has been a change in the paradigm in the way we look at microbiology today. So, as I go through my presentation, you will see that it, yeah, so we can first, I will go to my slides. I'll share the screen with you all. Just confirm that you can. Yeah. So, is my slide visible?
Yes, ma'am.
Yes, madam.
Yes,
It is
madam.
Okay,
visible.
okay.
Yes, ma'am.
So we are more or less set. So I just wanted to also to thank Dr. Shom, who primarily contacted me, and I was actually, in a way, happy to talk about this aspect because I thought you are already initiated to the field of microbiology, but as this, with the large number of bacterial genomes being sequenced and the problem of antimicrobial resistance lurking around us, I just wanted to take you through how this change is happening. And in between, while I like talk to you about the basics of how this change is happening, I will also sometimes bring in some research that we have done in the lab, and just to build context and to give you a reference of how things are happening. So I will start with my first slide, and can I can I do it from here? No. ওখানে যেতে হবে? Okay, that's better. But can I? Okay, so yeah, let's go to the first slide. So, see, whenever we talk about microbes, we know that not all of them are pathogens. Actually, most of them are not pathogens. They primarily are all around us in the soil, in the air, in water, and all of them, and also in our gut. A huge proportion on our gut, in our gut, and also on our skin. But none of them are actually pathogens. So I start my presentation by giving you a few things about what would you primarily think when you think that this is a pathogen. So we talk about the pathogenic personality. So it has to be a microbe which causes disease. These microbes will generally have some offensive and defensive factors. The offensive ones are like the toxins, sometimes the invasins, and all of this. And there are a lot of defensive factors like the siderophores, which help the bacteria to acquire iron in iron limiting situations. And also, uh, we know that this also has to be remembered that sometimes the distinction between the pathogens and the commensals can be blurred. The reason being that sometimes the ones which we consider as commensals, become, they pick up some genes from the pathogens, and then they cross the line and they actually become pathogens themselves. Okay. So, first, I come up with this WHO bacterial priority pathogen list, which was initially formulated in 2016 or 17, but there has been an update in 24, and this is the update that we have. And you can see that the World Health Organization has divided pathogens into three important groups: the critical group, the high group, and the medium group. And you can actually understand that the critical group are the ones which have uh, okay. So the critical group, as you can see here, do I have a mouse or something I can? Okay, if I move there, can they hear me? Okay, just a minute. I think a mouse will help me. Yeah, if you can see, so in the critical group, you have Acinetobacter baumannii, you have Enterobacterials, which are third generation cephalosporin resistant, and the carbapenem resistant Enterobacterials. Also for Acinetobacter, you will see it is carbapenem resistant. The Enterobacterials are actually the Enterobacteriaceae are now um, called enterobacterials, and they include pathogens like Escherichia coli, Klebsiella pneumoniae, and Olefins. So this group, you will see three of these pathogens are resistant to carbapenem, and you will probably hear a little bit from me about carbapenem, because a lot of the work that we do in our laboratory is on carbapenem resistant bacteria and Acinetobacter, Klebsiella, and E. coli. And these, when we talk about again resistance, this is an antibiotic which is considered to be the last resort antibiotics. And we will come to that when we talk about antimicrobial resistance. So you also have other bacteria in the high group and the medium group. Okay. Now, as I already said, Enterobacteriaceae or enterobacterials, Acinetobacter, Pseudomonas, all of these are very important or clinically significant bacteria. And particularly for the enterobacterials and the Acinetobacter, there are three different kinds of resistance mechanisms, all of which have actually pushed them and made them untreatable in certain settings, clinical settings, and hence they are considered critical. Okay. Now, you all know what antimicrobial resistance is. So, antimicrobial resistance is the reduction in the effectiveness of a drug when an antibiotic is not intended to kill or inhibit bacterial growth, then the term is used as resistance. So, whichever drug was earlier working against a group of bacteria have just stopped working against these bacteria, and hence they have become resistant to that antibiotics. And you can easily understand that if you are infected with such bacteria, then your treatment is a huge concern, because several antibiotics that were working earlier do not work now. Okay. So this, the left hand side of the slide actually shows the different antibiotic classes that were discovered and came into clinical use across these several years, starting with penicillin and then moving on to the one which I was talking about, carbapenems. In the later stages, that is after the discovery of carbapenems also, there have been other combinations of drugs, and India has also come up with a new drug called nafithromycin. If you can look up, you will see that that is also a new drug that has come in. But most of the earlier drugs are, bacteria is becoming resistant to them. Now, again, probably if you are, you already know a little bit on how bacteria can resist this different drugs. Just to give you a very brief idea, because we will also have to focus on the other aspects of these presentation. So the first is bacteria, most of them have efflux pump. So when drugs enter through, primarily through the porins, then there are these efflux pumps which can efflux out the antibiotic itself. You can also have certain enzymes like the one I was talking about. Carbapenems can be hydrolyzed by certain enzymes, or these antibiotics can be degraded by enzymes which are called carbapenemases, or there are also extended spectrum beta lactamases. So they will hydrolyze drugs like penicillin, ceftazidime, all of the beta lactam drugs. The other mechanism is modification of the binding site of the antibiotic. So an antibiotic was entering and binding and then killing the bacteria. Now that site has changed, and hence the antibiotic cannot work. Or there could also be other target alterations which can bring in antibiotic resistance. Okay. Now, one of lately, and a lot of work from my laboratory has been done on this, which is the New Delhi metallo-beta lactamase, which is also a carbapenem. And I have just brought in this to show that the presence of this enzyme is primarily making a bacteria or bug called a superbug, because such enzymes can actually hydrolyze several antibiotics and hence several beta lactam antibiotics, and also they carry along with them other genes which are resistant to other antibiotics. Okay, so now antibiotic resistance, as I said, is one of the major crises that the world is facing. And it is also called a, what would I say, a silent tsunami. So you cannot understand, but it is happening. It is nothing like the COVID, but it is quietly being there. And when you have someone who falls sick and is in the hospital, you know that an infection can happen, which is not cured by the different antibiotics that we have in clinical use. And this is the reason why the World Health Organization and also the World Health Organization has said that arguably the greatest risk to human health comes in the form of antibiotic resistant bacteria. We live in a bacterial world where we will never be able to stay ahead of the mutation curve. A test of our resilience is how far behind the curve we will allow ourselves to fall. So we are already behind the curve, if you can understand. And a lot of work across the globe is focusing on how to really bring in some changes so that antibiotic resistance is less of a problem. Now, again, a little bit of numbers or statistics, which, if you can see, 21.36 million people died globally in 2021 due to sepsis. And these cases of sepsis were not treated. Why? Because either the patient was very ill or the antibiotic did not work. Now, 4.71 million people who died in 2021 suffered from drug resistant infections. So these many people died because of drug resistant infections. And it is also predicted in an excellent paper by O'Neill. Initially, he said that 50 million people will die by 2050 if we do not make any change. But now there has been a recalculation of that, and it is said that 39 million people will die from AMR between 25 to 2050, and which is actually more than what people die due to cancer or diarrhea or malaria and all of that. Okay, so again, a picture of how grave the problem of antibiotic resistance is. And because you are all, in a way, teaching students, you can also, and it would really be good to bring up this topic and explain how the world is looking uh, now because of resistance. And all of this, anything in red you see, primarily denotes a warning sign where percentage of isolates with resistance is very, very high. So most of these countries, as you see, are in orange or red, indicating that resistance is very high. There is also something called the drug resistance index. And this index, again, if you see, India has the highest drug resistance index, which is 71. And this was calculated a few years ago. It could be more now. And this again shows that this is also a very, very big national problem. And I've just brought in this picture of Raghu Rai, one of the great photographers in India, of how huge the burden is for the nation and for the globe. So I have given you a little bit of idea of the problem of antimicrobial resistance. And I will again now go back and tell you about how there are techniques of identifying these resistance gene, how that was being done earlier, but how that is being done now with the advent or the progress of the sequencing of bacteria, the whole genome sequencing. So if you see, this was an era where Leeuwenhoek was grinding his glass lenses to look at something that he called the animalcules, and then came the golden age of microbiology, where Pasteur and Koch came up with their identification system, where biochemical identification was carried out. Then came the serological identification. Then came the automated systems like the Vitek and the Mardi. Then we also parallelly had um, Sanger sequencing, where the bacterial genomes were getting identified, but it took much, much longer time at that point of time. And now we have the whole genome sequencing and metagenomics. So this is a real paradigm change, I would say, and that is what is the focus of today's talk. So I will take you through how this process is changing. Okay. A little bit before I move on to the next slide is that even though we have come up with these newer technologies which give us a lot of data, we still, a lot of the clinical labs are using the earlier techniques also, because we have not absolutely shifted to these techniques primarily in diagnostics of antimicrobial resistance. And I will also take you through that. So this is a very simple slide where I have said that you will never know a microbe if you only look at the genome. So the point is, the genome, as in today's talk you will see, gives us a lot of information. It's huge. It's just humongous amount of information that is coming out, and it is coming out all at once. And but we have to also understand that not all of that, all of the information about a microbe can come from the genome. But as I said, a lot of that can come from the genome. So there has to be a balance between our understanding and sequencing genomes and also looking at other techniques, because they also add value. The reason being that the genome is like a blueprint. It gives you a lot of information about what is present in the microbe, but there are certain things which you know that the genes need to be expressed. There need to be expression of proteins and other things to actually manifest that what you see in that genome. Okay. Now, what are the major things that we can get from looking at the genome? The first is identification of the bacteria, and we will go in details through this. Then we can also know about the antibiotic resistant genes, the plasmid that these bacteria are carrying, the genetic environments. We know about the antibiotic resistance mechanisms, and a lot about molecular epidemiology. And all of this I will discuss now. Now, just to give in a context about the work that we do, most of the work that we do is primarily concerning neonatal infections because these vulnerable newborns have can have very quickly acquire pathogens, and they can become sick, and the neonatal sepsis is a huge cause of mortality and morbidity in neonates. And the treatment of these neonates for sepsis primarily depends on antibiotic resistance. So my work that we do in the lab, the the team that we have here primarily works on neonatal infections. We do a lot of work also in adult infections, but primarily antibiotic resistance in neonates. That is what we study. And our major collaborating institute is IPGMER and SSK Hospital. Again, a little bit on the neonatal world status, neonatal health status, and again, India has really done well by bringing down its mortality rate, but there is more to do, and one of the major impediments to that has been the problem of drug resistance. And again, you will see that India is a big country, and there are certain states like Kerala who are having very, very low neonatal mortality rates, but there are certain other states which have much higher rates. And as a country, we contribute to a huge burden of deaths in neonates. So, about 0.75 million neonates die every year in India. And a lot of that is because of drug resistance. Now we will move on with the bacterial genome part. So if you look into what we now call the next generation sequencing, so the sequencing of bacteria started somewhere around 1975-80, where primarily Sanger sequencing was being used. And this was a very tedious method, and hence, while this method was being used, you could see that uh, you could see that the number of genomes that were getting done were very few. But then you came up and come to this point around 2005, where we came up with this next generation sequencer, which is called the Iron Torrent. But now what has happened is the Illumina platform, as you will see, has also come in a little later, but this is one of the best platforms for next generation sequencing because the reads that you get are pretty accurate. And this, all of this has been categorized as short read. And lately now we also have something called the long read. So this phase between 2005 to 10, and now also we are using the Illumina machine. Number of labs are also using the Iron Torrent machine. Sanger sequencing is primarily being used to not to really look into the whole genomes, but to maybe look into certain genes. That is also being used, but not to do the genomes. If you have to look into the whole genome, then you primarily rely on the short reads in Illumina, and you also have the long read in the Nanopore and PacBio. And I will tell you how these two different reads are different. Okay. Again, how the advancement of these platforms have happened, and around primarily identifying the bacteria. So you will already see that when you come up with such different platforms, the identification of bacteria from the genome can become very, very easy. So, as I said, in this particular class, I will tell you about the techniques very briefly, because each of these techniques are very, very, I would say, heavy on the chemistry part, and I would not focus on that. I will primarily focus on how you can use that genome data for research, because these are techniques which can be taken care of by the machine. You only need to do a little bit of work in the lab, the rest is done by the machine itself. But you have to understand how you will use that genome data, and a little bit on the difference between the long and the short read. So, the short read gives you a depth of data because it fragments the DNA and reads it several times. So, the error in the short read is much less. But because you are fragmenting the DNA, sometimes between these two fragments there are certain gaps. The long read is primarily will read the whole strand of the DNA slowly, and hence there is what we call no, in terms of the gap, there will be no gaps, but the accuracy of the long read is much lesser than the short read, because in the short read, these fragments are getting read or sequenced several times. So, what we nowadays do is, for most of the work, we use the short read data, but sometimes for certain things, we also require the long read data, and we make something called the hybrid assembly. And this hybrid assembly is something which helps us to have a excellent picture of the whole genome. But for most of the work, you can really only do the short read and carry out several uh, of the things that you want to do. Okay, so as I said, there is a lot of these preparation steps which we do, but most of the work is being done by the machine. I will not go into the details of each of these, because that is a huge task which cannot be actually talked about in this class. So, my whole presentation now I have divided into these modules. So, we first will look into how genome data can be used for bacterial identification, and then we move on to the other things. And as we do that, I will constantly move and tell you about what was being done earlier, that is, what were the conventional methods, what methods were being used earlier, and how that is changing with the knowledge of the genome. But again, please do understand that we still use many of these in our labs along with the genome data. So the first thing is bacterial identification. As we had initially talked about, who is actually a pathogen, and if it is a pathogen, you need to know who that pathogen is, because how will you treat the patient if you do not know who that pathogen is? So either it's an E. coli or a Klebsiella or an Acinetobacter or a Staph aureus, you need to know who that pathogen is. So conventionally, we were using in our lab the biochemical test, and several labs across the world still use these methods, but this is a cumbersome method, and slowly we have moved from this to another automated system, which is not genome sequencing. But we will talk about genome sequencing after that. So this method is called the Vitek Compact 2 system by Biomeru. This we have in our lab and we use it regularly because we need to identify the bacteria that cause, as I said, neonatal sepsis, because we study neonatal sepsis. Now, the principle of this machine is basically the biochemical test only, but the system has automated this, so you do not need to make your media. The media are there in dried cupules in such uh, cassette like things, and there are 32 tests altogether, not 5 or 6. So they are there. You have to take, this is where you measure the OD of the bacteria, zero point 0.5 Macfarlane. You put it there, the machine, these cassettes will take in that bacterial growth, and then after a few hours, generally it is around 8 hours, but for slow growing bacteria it sometimes takes longer, and it identifies this bacteria. And you can see this is a typical result in from our lab, and you can see that this has been identified as Escherichia coli with a 96% probability, which is a very good match. So you know this organism is E. coli. Now that we are regularly using the Vitek, we also have at Nirbi, though I've written, this slide has NICET written, but the name of our institute now is the National Institute for Research in Bacterial Infections. I think I forgot to tell that earlier, and I'm sorry for that, but my slides, you will see that this is again a machine which is also present with us at the institute, and this is called the Malditov mass spectrometry. This machine again identifies bacteria. It has a large database, but it identifies not on the basis of the biochemical test, but on the basis of the protein signatures. So different bacterial species have different protein signatures, which is actually acquired by this machine, and then it is matched with the database, and finally you come up with a result. Again, Escherichia coli, the confidence score is 99.9%. And you can also see that this machine gives us, this is again a typical result would look like. So you have series of bacteria which have been identified. একটা ক্লাস নিচ্ছি মৌমিতা, পরে কল করি। একটু বলো, একটা ইয়েতে ক্লাসে আছি, ইনস্টিটিউটেই আছি কিন্তু আসছি তো। Okay. Okay, so now the identification of bacterial isolates using the genome sequences. So these are the methods, as I talked about, the earlier ones and then the later ones which we are using. But now with the genome data, you will see that once you have the genome, you can upload it to certain sites. There are several now. One of this that we use is the Pathogen Watch site, a global platform for genomic surveillance, where you just have to upload your genome data and you come up with the identity of that bacteria. And this one is, how is this primarily recognized? So the genome, you have a lot of data, and you will see each of them has some significance. But for identifying the bacteria is primarily done on the basis of the 16S rRNA gene, and also something which we call the multi-locus sequence typing. And I will talk about that later. in my slide. One more thing that if you have a bacterial genome, you will have to have a, you will, when you want to publish your research, before that, you need to submit your bacterial genome. The reason being that these genomes can be like, if you have sequenced a whole bacterial genome, you would have to place or put it into these sites so that others can also access it. And a lot of people are using this data for their own research. So if you go to the NCBI website, you have to upload your data. The data is checked by the NCBI portal, and then they give you a accession number for that genome. You can refer that in your, what would I say, in your papers or in your projects. And that accession number is a unique number for this bacterial genome that you have uploaded. Now, the next module, we will see that we have identified the bacteria maybe as E. coli or Klebsiella. But then, how do you characterize these bacteria in other ways? So, only knowing the species will not serve the purpose for a microbiologist, because the microbiologist will need to know certain things like what is the serotype of the bacteria, what is the sequence type of the bacteria, and you will see how this again is dependent on the genome. So in case of, this is an example of the E. coli phylotyping method. So all of these typing methods, you will understand, is because you want to actually give a kind of a signature to the bacteria. Suppose in case of E. coli phylotypes, you have uh, different phylogroups A, B1, B2, C, D, E, and F. And A and B1, these are primarily commensal E. coli, which you find in the gut. But this B2 is the ones which cause septicemic sepsis in or UTI and all of that. So this is the more pathogenic E. coli. So when you have this E. coli, one thing which you would like to know is what is the phylogroup group, because that gives you a kind of an idea of how pathogenic this bacteria is. So earlier what we used to do is, there was a conventional PCR based method. We also do it now. It's not that we can genome sequence all bacteria. That is still not what we are doing in the lab. So what we do is, on the basis of certain genes which are mentioned here, the presence of these genes bring up an algorithm, and you, whether this is present whereas this other one is not present, by such methods you distinguish whether it's an A, B1, B2 or D. And this is the PCR gel that we run, and we look at the different genes which are present, and then we make a decision on, okay, so these two genes are present, this is A, and that is not present, then that is B, and all of that. But now with genome sequencing, what can be done? You can take your genome and put it in a site called the Claremont typing, which primarily types the E. coli into different phylogroups. So Claremont is the name of the scientist who came up with this website, and all along you will see that there are uh, certain uh, softwares and certain websites where the genome can be read and meaningful data can come out from that. Okay, now as I was saying, you also, any microbiologist will know that serotyping of bacteria is very important. And in earlier times, when serotype was done in um, in the wet lab through the antibody antigen identification, we used to serotype bacteria. But now this serotyping is becoming less popular, and serotyping primarily is again now being done on the basis of the genome available. So this is the genome. This is another site, the Center for Genomic Epidemiology, an excellent site. We use it for several of our queries. And uh, these here, if you upload the data and go to the serotype finder, then the serotype will be determined and it will be present. So you can see here that on the basis of the presence of certain genes and the sequence of the genes, the serotypes are actually identified. So you have an E. coli which is O8H9. So that gives you a kind of a signature. So these are all epidemiological tools. Suppose you have an outbreak of E. coli and you want to know, okay, what serotype of E. coli is causing this outbreak? Is it an epidemic clone? Is it something more local? Does it match with other serotypes across the world? Then you will need that information. Okay. Uh, I think the MLST or the ST type, I do not have a separate slide, so I need to just go back and tell you about the ST typing, because that is also a typing tool. So MLST is multi-locus sequence typing. And just as you do a serotype, you also can do a multi-locus sequence typing. And this is like, it is a much better and a more critical tool for identification of the bacteria. And this is based on 7 different genes. You can see here, these are all housekeeping genes present in most bacteria. But for each bacteria, not only E. coli, Klebsiella, but within E. coli, so you can have differences in these genes even within E. coli. So E. coli again can be divided into on the basis of these 7 genes. And it is extremely important now as a very important epidemiological tool to identify. And suppose here this is an example of a ST131 E. coli. So you write ST131, which actually is an epidemic clone. So if you have this clone and you have this in a patient, you will know that this is that epidemic clone which has caused the infection. And with this epidemic clone is actually a lot of, what would I say? There is a lot of the ST131, suppose, is also very antibiotic resistant. So you also have a lot of information, and you can also compare your strains to other strains across the globe and find out in which countries are these strains present. And I will show you that later on. So this was one thing which was very important uh, in terms of, so there are different ways now to group bacteria. So you have phylogroups, groups, but within the phylogroups groups, you can have different STs. So you can see that each of these phylogroups groups again have a more distinguishing feature, which is called the sequence type. The other is the capsular type. For certain pathogens, particularly like Klebsiella pneumoniae, it is the capsular type is very important. Why? because again, that is a way you know that certain Klebsiella capsular types are much more virulent than the others, such as the K1, K2, K5, K20, and K54 and 57. So, all of these are uh, again, when you upload your genome data and you look for this tool which we use, the captive tool, this primarily gives you an idea of what K type this bacteria has. And this is how you get your result. So, this is a result from my lab. Different strains have different capsular types, and within those capsular types, you also can have different sequence types. So the sequence type, as I said, is much more granular. This gives you much more distinct features in the bacteria are picked up by the sequence type. So you have a K54 capsule type, you have another strain which is also a K54, but they are of different sequence types. But the capsular type gives you a gross idea of what the capsular type is. And instead of sometimes going into the ST types, the capsular type can also give you an idea of the virulence. Okay, now I come to the MLST part, which is described here. And you will see that initially, when we were not doing genome sequencing, what we were doing is we had these 7 genes, as I mentioned. We were sequencing these, we were doing PCR for these genes, and then we used to do Sanger sequencing for these genes, and we were uploading it to this Pasteur website. And these Pasteur website used to give different allele numbers, and a particular combination of an allele number used to give us the ST type. But now what we do is we either go to the captive site or the Cleborate site or the PubMLST site, which is a tool for determining the MLST. So wherever you do, whatever you do, the basic principle is based on the sequence of these 7 genes. But if you have the genome, you do not need to do an individual PCR to get the MLST number. You can just upload the genome because the 7 genes are present there in the genome. The sequence is there. The software will determine the MLST number. And here is again another site for MLST, which is the Center for Genomic Epidemiology. I have mentioned that earlier. So either you go to this site or you go to PubMLST. And as I said, there are several sites now, and many of them are very, very excellent sites being managed by some of the best researchers or research institutes in the world. So here you have, see, these are the different, these are the results that you have for this sequence type 131, which has been also identified as E. coli. Now, when we looked into all of these typing things, the simple question that might come to you is uh, what why do we require all of this? And this slide actually tells you why we require all of this. One is these typing methods help you detect emerging bacteria. So if you have, as I mentioned, an ST131, you know that this is an epidemic clone, it can spread, so you will be cautious. You also have a real-time monitoring of the transmission of these bacteria. So one outbreak could have happened in one hospital, but it can spread to others, or one unit of the hospital, it can spread to other units, and hence you can monitor. The other is you can always strengthen outbreak response based on the genomic surveillance data. Either you have the serotype or the MLST type, all of that helps you do that. The next and the last is you can also improve containment and prevention efforts. So that can also be done. So all of these in terms of medical science or is very important because all of this helps you either in prevention or in the response that you take when a disease occurs. Okay, moving on to the third module. The third module is looking into the virulence genes. And you will know that, as we know, and in my first slide, I have said that pathogens also have certain pathogenic factors. Certain of them are defensive factors like adhesins and fimbriae. They help them bind to certain surfaces. Others can be like toxins. So earlier we were primarily picking up our required genes from doing a PCR. But now we have changed our approach. We do this, but when we have the genome sequence, this is a site which is called the Virulence Factor Database or VFDB. But there are other sites like, as I said, the Captive and the Cleborate. All of that also gives you an idea of the virulence factors. And this is a data from my lab. These are the different isolates, and once the data was uploaded for all of these isolates, we could see the presence of certain genes. And these are the genes which were important for identifying the virulence in certain Klebsiella pneumoniae. And if you look into these, we looked into the different lineages of these genes and the presence of the genes. So at one shot, you now have the idea of what is the bacteria. You have an idea of what is the serotype or ST type of that bacteria. You also know what are the virulence factors. Again, the CGE site also gives you something. There is something in this site called the virulence finder. So the earlier one was a VFDB site. Then you have a virulence finder where you can actually read the virulence genes. This is again an example of the heat map where the virulence genes are indicated. Now we come to what is primarily carried out in my lab in terms of antimicrobial resistance, because we do a lot of work around antimicrobial resistance. So we still do a lot of phenotypic tests. You know that these are disk diffusion methods using different antibiotic discs. We also carry out something called the E-test for determining the MIC value of the bacteria. We also do all of these like the microbe broth dilution and all of these. We also look for certain genes, antibiotic resistant genes in this bacteria, but what we now tend to do is, if we have a genome sequence, you can just upload that and you have something called the res finder. Res finder is something which stands for resistance finder, and there is also another software called the card. All of these give you an idea of what are the resistant genes present, and it is not one or two. So you can look, because you have the genome, you have a whole idea of what are the resistant genes against beta lactam antibiotics, against fluoroquinolones, against tetracycline, against carbapenem. So all of that data comes to you all at once, and you can see that this is how the results are depicted in these sites. And they also give you an idea of which antibiotics this particular bacteria is resistant to. So these earlier methods that we were doing, as I said, We still continue to do that. Primarily, we do the broad dilution method, but this MIC value we not only do by the broad dilution method, but we also get results from the Vitek machine, which I had earlier shown that the Vitek is also a tool by which you can identify the susceptibility pattern. So we do that, and we also sometimes rely on the whole genome data. And this is again data from my lab, and this is from a paper that we've already published, and you can see that there are so many resistant genes in different categories which are marked here, which are present in these isolates, and all of these have come from the whole genome data. Now, apart from looking into the resistant genes, what else does the whole genome data give us? As I said, it gives us a lot of information. So earlier we used to look at, purify the plasmids, look at the size of the plasmids, and understand through conjugation experiments whether these plasmids can be transferred. But now look into this. This is a map of a plasmid. So you have the genome data, and the long read helps you along with the short read to create such maps of the plasmid. And you have, and the plasmids that we have in the uh, clinical isolates, the isolates that cause sepsis, are very huge. These are large plasmids. They are like 120 kb, sometimes 200 kb. All of these are present, and there are several genes. You can see from the genome data these different genes are present. And this is a plasmid which had NDM. I was talking about the presence of NDM, which indicates the presence of a superbug. So this NDM was present on these plasmids. These are different plasmids from two different bacteria. But these plasmids, again, are different, or there is something called, as you have typing for the strain, the ST typing, you also have the typing of the plasmids, which is called PMLST. But apart from that, we also require to know the ink, the ink type, or the uh, replicon type. So the replicon type is also again another signature for the plasmid. So here you have a type which is called the Y, whereas here this plasmid is FIA and F2B plasmid. And this plasmid, from several of our studies, we have noted is very common in E. coli and Klebsiella, and other researchers have also noted that. So when you get these plasmids, you can also compare these plasmids to other plasmids. And this is a picture of the PNDMR plasmid, which was assembled in our lab. And you can see these maps in the plasmid. Okay, so as I mentioned earlier, antibiotic resistance genes or were identified through PCR. We also identified the what would I say, the just now I mentioned something called the ink types or the replicon types. We used to identify these replicons through a particular PCR based replicon typing method, which was again based on the PCR. But now, as we have the genome data present, we just have again go to the Center for Genomic Epidemiology site. We have something called the plasmid finder. So you had something called the res finder for resistance gene. You have something called the plasmid finder to know which are the plasmids that are present. So again, the genome data is helping us with the plasmid. And as I said, as you have a signature for the bacteria, you also have sequence type of these plasmids. Again, this is an epidemiological tool because you know that in some cases, certain plasmids can spread very quickly from one bacteria to the other, and certain plasmid ST types have been recognized, and this is one type. You can see it gives us a very, very detailed data of what is the F type and then whether that is A or B1. All of that is given to you. Something that we also do, as I said, in clinical settings, and this is again a data from my lab, is comparing plasmids. So we had a study where several of the pregnant mothers had a particular bacteria MCR positive E. coli present, and we wanted to see were they same, and we compared the plasmid sequences, and you can see that they were more or less same, except a few this wide gaps are the differences that you had. So that also gives us an idea of how the plasmid is spreading. The other thing that we know is about the genetic environment. So, if you have a particular resistance gene, you want to know what is upstream and downstream. And this also comes from the genome data. So, as I again coming up with this particular gene NDM, this has a particular IS element or the insertion sequence which is very typical of this gene. And then this gene has in its downstream the BLE, MBL, TRPF, and all of that, again an IS element. And this kind of genetic environment is seen in many isolates, and this is also something we study because we want to understand what are these sequences that are present upstream and downstream of a particular resistance gene. Okay, coming to the sixth module, and one of the most important modules, I would say, and I have talked about that, and I will talk again because we will use those principles that we have studied to understand how we compare strains. So, as I said, when you first have a genome, you can upload that genome to the NCBI website. So, your genome gets a unique number and it is present in the NCBI website. So, several genomes are present in the NCBI website, either from different countries, US, Japan, China, Taiwan. Name a country, you will have sequences of bacteria uploaded in that website. Now, there is sometimes a requirement of knowing whether our strains match with their strains. And as I said, one way of knowing is to look at the sequence type, the serotype, but we also sometimes do something where we compare these genomes and see how close they are. But before I go into that, I would just like to tell you that before this happened, or when genome sequencing was not done, a lot of tracking of infection sources were also done by a method called the pulse field gel electrophoresis. So the pulse field gel electrophoresis is a method which is a gel electrophoresis method, but the whole genome is digested by an enzyme, and then it is actually these gels are run in a gel apparatus where the direction of the current constantly changes. Like you will have a 30-degree, the current will move in a 30-degree angle, then in a 60-degree angle, and in such a way the fragments get fragments get differentiated and you can compare the strains. We, again, I would say we still use that in our lab now, and these are very good to track small outbreaks. But you cannot compare, if you see these are different isolates from a particular study, all of these are from different patients, but they are the same isolate. Again, you have another type which is again different from these, but they are same in these patients. So we can understand which, by these patterns, which bacteria are same. But you have to understand that with such data, you cannot compare with isolates in US or Japan and Taiwan. So the genome data here again is how we compared our strains through a particular website, and these the gels can also be compared through different softwares. This is a picture of that. But what is more important is that now with the genomes, you can look into your strains across several years. Suppose we are studying neonatal sepsis for the last 20 years. We want to know whether certain strains in 2008 match with the strains in 2018 or maybe 25. We can compare these genomes and see how close they are. So you can see when you have something like this, 100% match, you can, and this is not, this is from the genome data. And that is the reason we call it a core genome phylogeny. So based on the core genome of the bacteria, you carry out these kind of analysis to know which are close to each other. But this one is a core genome phylogeny of our strains, but also strains from other countries. So you see the list of the countries here. We went to the NCBI website, we downloaded strains of Klebsiella pneumoniae present in different countries. Suppose we want to look into Klebsiella pneumoniae causing neonatal sepsis. So we look into the database of different countries, we download these sequences, we have our own sequences, and we compare these sequences, and you can see that you can also compare so many genomes together from a you can compare these genomes with your genomes across the globe, and this data is also very important. And suppose you, as I mentioned, the ST131 isolate. So this is again a study from my lab where several of the ST131s are ST131s, and those from other countries were also compared. And you can see how these match with each other. So you can compare and see how far your ST131s are from strains from other countries and how they are close to them. And you also have something called the single nucleotide polymorphism, which gives you how farther off from one strain to the other is. So that gives you also idea. Again, this was another example. Uh, I will quickly give you an idea. Again, we have maybe 5 to 10 minutes of time. So, as I said, antibiotic resistance, you have a number of genes, one of which is the efflux pump genes. And this is one particular study that we did where we looked into different efflux pump genes. And we looked into how the expression levels of these genes were changing. And we wanted to know, why is the expression level changing? Again, we earlier what we used to do is we looked into each gene, we carried out PCR, we carried out Sanger sequencing, but now you have the whole genome, And as I said, you can look into the the mutations in the different genes. And in this study, what we did was we had certain of our isolates with mutation in this gene called MGR, and we downloaded other sequences, global sequences, and we compared them, and we wanted to see whether those isolates were having resistance, having mutations or not. And we found that several of those mutations were similar to the mutations that we found. And with this kind of data, we can also do structure prediction. So, this was a study where we saw the mutations in porins, and we could, with the use of softwares like PyMol and all of that, I my lab does not do much of this. We do it with the help of our collaborators who do a lot of take our sequences and do structures and give you predictions of why the change in the structure is causing resistance. So here again, you require the Cluster W website, where you compare your patterns with other patterns, you see where the change has happened, and then you do the modeling. So you have a basic model of this porine protein, but you also show how the loops are changing, what is happening. And this study showed that the change in the loop structure, actually the porine has a barrel shape through which the molecules enter through one side and are passed on to the other side. But with certain mutations, this barrel gets restricted because the loops are moving into the barrel, and we understood why the resistance was happening because of the change in the structure. So this can also can be done from the genome studies. So, what I wanted to tell you and give you an idea was that the genome sequence has become an excellent tool for the world to identify bacteria, to find out certain signatures like the sequence type or the serotype for the bacteria. It gives you information about the resistance genes that are present. It gives you information about the plasmids that are present. It gives you information of whether your strain is an epidemic strain. It tells you whether you can compare your isolates with other isolates and see how far they are, how close they are. You can also look into certain genes, mutation in those genes, take them forward to understand the structural changes which can lead to overexpression of genes, resistance. You can see why a certain bacteria is becoming resistant due to the change of certain mutations, and all of that can be done from the genome sequence. But what we understand, but what I want to reiterate, which I said at the very beginning of my presentation, is that genome is one part of your tool, but do not forget to use the other tools as and when required. Of course, you will use tools which are quicker, and the genome sequences or the Vitek and the MALDI, all of these are quick methods of identification. But the genome is still not used in clinical setups, but several hospitals across even Kolkata and West Bengal have the Vitek machines. Even the government hospitals have the Vitek machine because it is such an easy machine to identify these bacteria and also know the susceptibility pattern. But the genome sequence is also important in several ways by giving you a exhaustive amount of data, a lot of the data as we discussed today. But something that I also tell my students is that, particularly when you are doing research, I like to take this quote from Bill Watterson, who said that your preparation for the real world is not in the answers you've learned, but in the questions you've learned how to ask yourself. So you may have the tools, but it's tools which, and you know these tools, you know how to use these tools, but you have to have questions, the real questions that you need to ask to solve a problem. And that question does not come from the tool. The tool can only give you the solution. The question will come from your understanding of the problem, and by understanding that question, you will know how to use these tools to find out about uh, the solutions of these problems that you see. So, before we move into the interaction session, just want to uh, acknowledge a lot of my students here, little bit of work from many of them, and this depiction is also uh, mm, done by my students, our lab at Nirbi. But I would also like to thank several clinicians, particularly from IPAGMER and SSKM Hospital, with whom we've been working for the last 20 years. We have also started working with other medical colleges, but with SSKM, we have a long term collaboration. I would like to thank Dr. Rajlakshmi for her contribution in some of our work, Dr. Shogato Hajra from IIT Roorkee for his work with the structures, then some of the work which we did with Cardiff University and the data analysis by Kirsty and Dr. Purnoslok Bhaduri. And I would also like to thank the different funders for the work that we do. And so I would also like to just bring it one more thing, which is that we have a bacterial repository at Nirbi, and through this repository, we can share strains with researchers. So if you write to me, or I can even share the website with you, we can you can also have access to certain strains which you may need for your research work. So with that, I would like to end my presentation and would look forward to questions that we have. Thank you.
As we conclude this enlightening seminar, I would like to express my heartfelt gratitude to our honorable speaker for sharing her immense knowledge and expertise with us by providing a complete blueprint of an organism's genetic makeup. Genome sequences allow researchers to understand the evolutionary relationship, ecological roles, functional capacities of bacteria with unprecedented precision. Overall, bacterial genome data serve as a foundational tool that strengthens scientific discovery, improves health outcomes, and foster innovation across multiple sectors. Now, session is open to address your doubts and queries. Dear participant, please feel free to ask a question by raise your hands, and in the chat box, Madam will address them.
I think there was a lot of material in this session, but it was primarily not to scare you off, but to give you a little bit and to raise a kind of awareness about uh, raise awareness about AMR and how we use bacterial genome these days for our work, and also how the whole what would I say, the field of microbiology is changing with this. So
Madam,
if you have
there
any
are
specific questions, yeah.
uh, from Navanita Ghosh, E. coli is one bacteria which have in our gut healthy condition as well. Then when does E. coli become pathogenic in its effect strain specific?
Yes, I understand your question. You are very right. E. coli is one of the first pathogens that will actually colonize the gut after the birth of a neonate. And most E. coli that we have in the gut are actually commensals. And as I said, the if you divide this whole species into different phylogroups, I mentioned in one of my slides when I was talking about phylogroups that the A, B1, these are primarily commensals, but B2 and D, these are primarily the pathogens. And why they become pathogens is because they acquire certain genes, and these genes are either, we have a lot of genes in E. coli. We have a lot of something called the serine proteases or spades, which some of which are virulence factors. We have invasins, we have siderophores, we have fimbri, all of these factors, and sometimes toxins. Like, suppose enteropathogenic E. coli has certain toxins, enterotoxigenic E. coli has certain toxins, and there is the whole E. coli group is again divided into something called the, among the pathogens also there are groups, like some of these are gut pathogens, so they are called intestinal E. coli. But there is also a part called the extraintestinal E. coli, which causes infections like sepsis, UTI. A common cause of urinary tract infection is E. coli, and these E. coli which cause sepsis or UTI are having certain virulence factors, due to which they are different from the commensals. And these factors, they acquire either from other bacteria, as has been seen that sometimes they have acquired it from other bacteria, or they one particular gene has been shared through plasmids between different E. coli, and that also makes a commensal E. coli become a pathogen. But certain types are actually, as I said, uh, if you look into the broader evolutionary tree of E. coli, you will see that certain phylotypes like B2 and all are actually the pathogenic types. They spread their genes to others, but you can also have, like, these can also spread because they have epidemic potential. I think I
Thank
have.
you, ma'am. Another question from Arijit Dash. Are there specific conditions where a good bacteria coming in contact with pathogenic bacteria itself becomes pathogenic, or the event is random? Can a resistant bacteria convert other surrounding bacteria resistant?
Yes, you are very right. I should have delved more into this. See, there are several resistance genes which have been initially identified just from soil bacteria and all of that. But these got selected with the overuse or misuse of antibiotics. So, as you say, if there are certain resistant gene in a bacteria, the bacteria may spread the gene without any reason. but it has more reason to spread the gene or acquire the gene from others when it is in a condition where antibiotic use is very high. And that is the reason why the spread of such bacteria can happen in the hospitals where antibiotics are used heavily. So, suppose there's a bacteria, it is a basic survival instinct for the bacteria. The bacteria, there are two different bacteria present, one with the resistance gene, other without the resistant gene, and you place them both in an environment of antibiotic. The one that does not have will, because it wants to survive, will actually acquire these genes, and many of these transfer take place in our gut also through something which we call the process of conjugation. Conjugation is a method where you can the like one bacteria can transfer its genes to the other bacteria by this process, but there are other processes like transformation also, but conjugation is primarily most important. And we have seen in the lab also, under laboratory conditions, the use of antibiotics will actually help the spread of these genes. But again, the use of antibiotics can also create mutations in certain genes, and in that way also bacteria become resistant. So they can acquire genes, but in the presence of antibiotic uh, they can actually have mutations in certain genes, which also brings in antibiotic resistance. To yes, they generally require a trigger, but sometimes you will, because a plasmid bearing these large plasmids also bring in a fitness cost to the bacteria. So it will only require that when the environment is such that it really wants to live and does not have the genes to cope up with that environment. And that has been seen with the transfer of virulence genes also. So virulence genes can also spread in certain conditions when the bacteria is under stress, where the bacteria has iron limiting conditions, then you can see the transmission of siderophore genes when the bacteria is. So that is.
The last
Somebody has a question,
question.
what role does a human microbiome has? I think that's a question which somebody asked.
Madam, last question in the chat box. How helpful is NGS in identifying new species? Do you still need to analyze cell wall composition, DNA hybridization, etc., even after performing whole genome sequencing by NGS?
Yeah, so for newer species, uh, you NGS is a very important tool, but before you call that a new species, you also require the other some of the other conventional methods which we use. But nowadays, a lot of work is focusing on only the use of the NGS, and then what would I say, corroborating with known data which is present in these freely available sites, and then you have to report them, and then it is identified as a new species. But to be very honest with you, we do not work on new bacteria, nor do we work on microbiomes. So that part of it, and in clinicals, because we do more on pathogens and the clinical, we use bacteria from the clinics. Here, the genomes are primarily known, so these are not new genomes, and we do not find any difficulty. But I can come back to you and let you know. But I have certain colleagues, not in NIRBI, but other collaborators who work on environmental bacteria, and they do get a lot of new species and they primarily depend on the NGS data.
Dr. Arunima Das, please ask your question by unmute your voice, mic.
Thank you, madam. Uh, Madam, it was a very detailed description of how bacterial infections are caused and how we are trying to combat it. Your work, you have said, that is restricted, or maybe you are basically working on neonatal sepsis. So there are two conditions through which the childbirth is taken care of. One is through the vaginal birth, and the other is by C-sections. Now, there has been various studies that show that if vaginal birth is preferred, then the neonate actually gets more beneficial bacteria that enters into its gut, and it is better for establishment of a good flora. But in case of C-sections, the child is deprived of that. So is it more prone, or the children are more prone to sepsis if they undergo vaginal birth, or they are more prone to sepsis if they are being delivered through C-section?
No, the risk factor for sepsis generally is not the mode of delivery. There are other several factors for the occurrence of sepsis. Suppose a child is preterm, is born earlier than 36 weeks, or has a very low birth weight, gestational weight is very low, and if it has certain congenital malformities, the chances of sepsis is much more, because the immune system of such neonates is compromised. So that is the primary reason for sepsis. The mode of delivery, as you say, and certain of our studies have also shown that during vaginal birth, the bacteria from the mother's vagina or the rectal swabs that we take um, does have can move on to the child. But see, what we have also seen, that if there is no antibiotic pressure, these organisms, even if they move on to the child, will not cause subsequent infection, uh, because the child is immunologically absolutely fine, and these bacteria do not are not retained for several months. So once the baby is out of the hospital, if we take samples within a few months, we find that that bacteria that we had seen earlier is not there. So the reason or the risk factor for sepsis is primarily not the mode of delivery. Yes, several, there are actually much more advantages of the vaginal delivery, both for the mother and the child, and conventionally that is the mode which has been followed. But we know these days that there is a huge amount of section deliveries, and that also introduced, because it's a what would I say, it's a surgical intervention. So what happens is that the mother again becomes introduced to resistant bugs from the hospitals in the OT and all of that. So that can also become a cause of infection for the child. So mode of delivery is not the primary risk factors for sepsis. A mother having infection earlier, a UTI, or some other problems like premature rupture of membrane, the baby being low birth, and as I mentioned, premature, all of that are the risk factors of sepsis.
In case of premature deliveries or preterms, the mode of delivery is C-section or natural birth? Because natural birth or vaginal delivery will
No,
have.
no, no. See, the decision on doing a C-section is absolutely up to the clinician, and sometimes also these days the choice of the mother.
Okay.
But there are cases where the mother has a premature rupture of membrane and there is a chance the baby may get an infection very soon. In these cases, an emergency C-section could be done. But a decision on a what would a planned cesarean section is something that is done between the mother and the clinician. It has nothing to do with the baby. There are certain complications due to which you also require C-sections. So that is a medical requirement. But a lot of the C-sections are just being done out of choice, I would say. And it has nothing to and that does not determine. As I said, the mother may have a premature rupture of membrane at about maybe 30 weeks. The baby is very small, the doctor has nothing to do but to deliver the baby. The baby is then kept in the neonatal intensive care unit for a long period of time. And the presence of the baby long term in a hospital is not good for the baby with certain devices. You can have a ventilator on the baby, you can have a catheter. There are several invasive devices on the baby which also increases the risk of infection. So all of that are there. So it's not, again, I would say it's not the
Primarily the delivery
right.
pathway,
Yeah, yeah, yeah.
but it can be, and the hygienic conditions of the NICU should also be taken into
Care
consideration.
of the hygiene conditions of the mother, the exposure of the mother to antibiotics. We have seen that is also a very big factor because a lot of mothers and a lot of people, I don't say mothers, take antibiotics indiscriminately, which actually changes their gut flora and gives helps in the colonization of resistant bacteria, which can then be passed on to the babies. So that is also something we need to. Today, my talk primarily focused on bacterial genomes, but sometime, if there is an opportunity, I would like to talk about the problem of antibiotic resistance per se and how this problem is escalating and the several factors around that. So I think that would be a better
Of course.
way of, yeah.
Right.
Madam,
Thank you, ma'am.
due to shortage of time, we need to wrap up. I
Yes.
appreciate all attendees for their active participation and valuable contribution. It's been a pleasure hosting this online refresher course, and I hope everyone leaves today with a greater understanding and new perspective on our focus topic. Thank you, Madam and participant, for making this seminar a success.
Thank you. And I have shared the handouts of the slides to Dr. Shom as
Okay, ma'am.
requested.
Okay, thank you so much, madam.
Thank you so much. ঘরটা ভীষণ ঠান্ডা হয়ে গেছে। ওকে, এটা দিয়ে দিয়েছে, এটা দিয়ে দিয়েছে। হ্যাঁ, করবো। জি।
Good afternoon, everyone. We'll be shortly beginning with our second session of this program, which is the online refresher course on bioscience, bioengineering, and nanotechnology. Our esteemed speaker, Professor Dr. Shoykot Mitra, has hopefully joined. Hello, sir. Have you joined? Okay.
Hello. Am I audible to all of you?
Yeah. Yes, sir. Okay. I, on behalf of Maulana Azad College, welcome our speaker, Dr. Professor Shoykot Moitro, and all the participants to this program. Before we begin with the lecture, let me introduce Dr. Shoykot Moitro. Professor Dr. Shoykot Moitro is a distinguished academician, scientist, and administrator who has formerly served as the Vice Chancellor of Maulana Abdul Kalam Azad University of Technology. Prior to this, he was a faculty member at University Technology Petronas, Malaysia, and held major leadership roles such as professor and principal in charge at the Government College of Engineering and Ceramic Technology and at RCC Institute of Information Technology, Kolkata. Earlier in his career, he worked as senior scientist at the Central Fuel Research Institute, Dhanbad. He currently contributes to state-level academic governance as chairman of the West Bengal Artificial Intelligence Task Force, an expert member of the State Education Policy Committee, among other roles. Born in 1964, Professor Moistro holds multiple degrees from the University of Calcutta, including BTech, MTech, and PhD in chemical technology. He also has a PG diploma in management and is a certified chartered engineer in chemical engineering. He has completed several international specializations in management, technology, and education. If that was not all, he is also a prolific researcher in chemical, material, and environmental science and has 160 publications in SCI index journals, presented 50 conference papers, and authored 7 books or book chapters. His research impact includes an H index of 30, I-10 index of 53, and 3,524 citations. He holds 2 patents and has completed 11 funded projects. Professor Maitra has received 9 awards, including the prestigious S.S. Bhatnagar Award conferred to him by the Indian Science Congress in 2019, and he is also a fellow of the Institute of Engineers, India. He is an active member of several national, scientific, and professional bodies and has delivered lectures worldwide, including the United States of America, United Kingdom, Australia, Italy, Malaysia, and others. After such an introduction, I think that all the participants are eagerly waiting for his lecture, and I hope that all of you take back lots of memories and experiences and whatever lessons he has to deliver. And please let me remind you that the lecture session is about of 1.5 hours. After that, there will be a Q&A session where you can ask your question. Thank you, sir. Moving over to Dr. Soikot Moitro. So you may please do the honor of beginning with our session. Thank you.
Thank you very much. Let me share my screen first. Is it visible to everyone?
Yes, sir.
Okay. Okay.
Sir, any problem with the presentation? Can I help?
The participants are requested to wait. I think that our speaker is facing some issues with his presentation. Our session will be beginning shortly.
Can you see the screen now?
Yes, sir.
Yes, sir.
Okay, let me start my presentation. Basically, I would like to extend my heartiest thanks to the organizer for inviting me and for the nice introduction. Now, the matter of fact is, I did some work on quantum mechanics, and from a long time, I'm trying to understand what is quantum mechanics. And of the late, particularly in this decade, quantum mechanics now are trying to, these come in practical application in the form of quantum computing. And I'm trying to collate all these experiences and understandings which I'm having in quantum mechanics in every aspects of its applications, starting from cosmology to this material science to life and everything. And it is a part of that. I'm trying to write a book on all these things, and one chapter has been dedicated to applications of quantum mechanics in life. Although it's not new thing, because long time before, you probably have heard this name of Erwin Schrödinger, who is famous for this wave equation, and which and from his equation, this dawn of this quantum computing, quantum mechanics started, and there afterwards he has written a book, What is Life? And life is nothing but quantum mechanics. So everything is happening in this in atomic levels, and maybe in subatomic level, electronic levels, and the waves of all these these subatomic particles, and their nature, their coherency, and their the entanglement, their superpositions, everything that determines the fact how everything will propagate. So. Here, I'm trying to, this correlate, some areas of modern bioengineering application in with regards to cancer treatment, and how quantum mechanics, quantum computing rather, can play a big role in it. Probably you are aware of the fact that some organizations, there is tough competition is going on. Who would be the first launcher of quantum computer? And so far, my information goes, IBM now is leading this race. Of course, there are other organizations, many countries, they are fighting for this race in US, China, UK, Canada, France, all these advanced countries. IBM is leading, followed by these Googles and others also. They are also coming in the picture. And maybe by 2030, who will be watching the launching of commercial versions of quantum computer in the market. Now, this regarding this cancer, if we look in the history, in 1971, President Nixon, Nixon is famous for what not so famous for many things. He announced the war on cancer and declared this modern medicine would finally end these scars, because that time certain these positive results of traditional cancer treatment that came out, and he became over enthusiastic and declared that now people would not suffer from cancer. We have this we have understood the treatment for the different types of treatments to eliminate this disease from the human race. And he allocated lots of funds, research funds, for this cancer treatment at that point of time. Nixon is famous in this, not so famous in that sense. He was instrumental in 1971, this Indo-Pakistan war involving Bangladesh, and he supported Pakistan. That's a different story. But this fellow, he allocated or allotted significant amount of funds for cancer treatment, and this traditional treatment, surgery, chemotherapy, radiation, these are traditional treatment of dealing with cancer, and that for intensifying research in these areas. But what happened afterwards? What is the result? Result is not so significant, because the improvement was incremental. Because the traditional treatment, it is targeting on this probability. This is also quantum mechanics, and this is this positions and this velocity. These two cannot be precisely determined. Here happens these targeted cells where they were assuming that cancer has happened. This is a probabilistic scenario, most probable area, but that doesn't mean in other areas cancer have not percolated or spread. So cancer still now is the second leading killer in US after this cardiac arrest and all. But and India also in 2024 the figure says there's a significant amount of cancer death. And here there are many types of cancers. Most common cancers in from the data from different hospitals, we have seen that the oral cavity cancers, because we Indians have a habit of chewing this pan muscle and this brittle and all this the brittle nuts and all these types of these things which are not so healthy for for oral health. Lung cancer, stomach cancer, these are the most prevalent cancer in men, and in women breast cancer, cervical cancer and ovarian cancer are the most prevalent types of cancer. Now, what is the fundamental problem? Why after 1971, these huge, these efforts on this cancer research that did not give these targeted results? Because that the reason behind it, what is actually cancer? That time there is a debate, raising debate, and for example, there is a debate regarding quantum mechanics also at one point of time between Einstein and Niels Bohr, whether quantum mechanics is the real science or it is pseudoscience. That types of this debate went on, but ultimately we know that quantum mechanics is a reality. Similarly, cancer. What is cancer? That time there was a difference in opinion. Many people, they were of the opinion that cancer is caused by a single factor, or sometimes they are of the opinion that it's combination of diet, pollution, genetics, virus, radiation, smoking, or just a probability, bad luck. So this was the debate. Now, afterwards, when our genetics and biotechnology it advanced, we understood these are gene level many things, and we understood the applications of these biological processes in technological applications. Then we understood that at fundamental level, cancer is a disease of our gene. Everyone maybe this probability of having certain sorts of coding at our gene for cancer, but it can be triggered by different environmental poisons, radiations, other factors, or sometimes probability, bad luck. So cancer is not one disease, because thousands of different types of mutations is happening at our gene. And sometimes some mutations, these are, developing bad results. Now, there are so many varieties of cancer till now. Now, there's an encyclopedia of the various types of cancer that cause healthy cells to suddenly proliferate and kills the host. Yesterday is benign, today malign. So this is the problem with cancer. Now, if we look into the different types of cancer, the different types of cancer include carcinoma. Carcinoma basically it is a skin and organ linings is affected. Sarcoma is a bone and soft tissues. Leukemia, cancer, it is related to blood and bone marrow. Then melanoma basically develops from pigment producing cells that is called melanocytes. Then the lymphoma, that is lymphoma is the lymphatic systems. This is different types, but there are other types of categorization also. For example, where it is located, prostate cancer, breast cancer, colon cancer, lung cancer, etc. It can be broadly divided into categories. One is a solid tumor, another is this blood or hematologic cancers. So there are different ways of classifying cancers. Now, this cancer is incredibly diverse and pervasive disease. It started with this after this human evolution or human race came in this in the world. From that time, because this oldest medical reference of this cancer, it went back to 3000 BC in Egypt. In the mummy, we found this, these are the symptoms of cancer. Before that, we do not have any evidence. It's prehistorical ages, but cancer was there for, and cancer it is found in animal kingdom, in dogs, in cats, in all sorts of animals. You can find. I am a dog lover, and I became instrumental in treating some dogs, the stray dogs for this cancer a few days before. But this is prevalent. This disease is for every living system. There is a probability of cancer. Now you see the term called apoptosis. Apoptosis basically it is for creation of a complex life that involves trillions of cells. This earlier evolutionary biology tells life started from the single cell, and then it becomes this more and more complicated with the formation of more and more cells and all this. And trillions of cells is required to form a complex organisms. And these trillions of cells, it involves complicated chemical reactions in sequence. Now, for giving birth to new cells to come, old cells need to die. This is the mechanisms. Old should go, and new will come. So this is the natural phenomena. Now, sometimes this is not happening. Old cells, they are resistant. They do not want to die. So this apoptosis process, if we look at it, there are different steps involved. There is initiation. In the initiation, a signal is received either from internal stress or external triggers that activates the entire pathway. Then after that, there is execution. Execution is this entire stage is execution. This execution involves the shrinkage of the cells. Then chromatin condensation and DNA fragmentation that happens. This material inside the nucleus condenses and breaks into fragments. Then cytoskeleton outside this part that collapses, this internal scaffolding of the cells that breaks down. And then a membrane blebbing takes place, the cell membrane from bubble-like, this protrusion, and then formation of apoptotic bodies. The cell fragments into small membrane enclosed vesicles called apoptotic bodies, and ultimately phagocytosis effect is there. The phagocytosis that is called macrophage. These are macrophage examples. This engulf and clear the apoptotic bodies. It is for last for the cleansing. So cells are genetically programmed to die by necessity, sacrificing themselves to create new complex tissues and organs. This is the thing. But although this is the program, but sometimes there is an aberration. There is this bug in the program. And error in the programs. Error sometimes this natural process is turned off, and the cells continue to reproduce and proliferate instead of being dead. That means cancer cell is immortal. it doesn't die. This is the fact Mother Nature has taken care of. That otherwise our this length, our everything would have been this this significantly, or this up to maybe infinite, if we continue to grow and grow and grow and without dying. So that there is a limitation of everything. The upper limit, upper bound was placed on that by these dying mechanisms. But this cancer, what is happening because of this programming, this change in this coding and programming at this molecular, at this genetic level, and at the molecular level, at the subatomic levels, because of this change in these wave patterns, all this. This is also again quantum mechanics why this cancer is happening. And then this ultimately it is converted to double and then cell division, then malignant cancer cell is formed. And these cancer cells, they basically, what they do, they grow uncontrollably and they create tumor, and that eventually shut down our vital bodily functions. So that's why we die out of cancer. Now, cancer formation is a peculiar thing. It often takes many years or decades for cancer to form. Maybe if we have a sunbath at early stage of a life, at childhood stage, if there is a sunburn, we may get skin cancer at that very spot, but years later. Because this requires more than one mutation. As I already told, that more than 1,000 mutations is required, so more and more numbers of mutations. One triggered point has started there, but it continued and continued over the year. There are time factors. And after that, maybe after many decades, this cancer is formed, skin cancer. So this basically, finally, it is they disable the cell's ability to control reproductions. If we look at the normal systems, these normal cell systems, but when there's a cancer cells, these cancer cells, you can see its nucleus becomes bigger and darker than normal, and shape becomes a non-uniform. That's why this propagation tendency and proliferation tendency is more because of this different sort of protrusion and different sites. Abnormal number of chromosomes arranged in disorganized functions, and disorganized means randomization, and this creates chaos. Chaos is another part, and then this creates clusters of cells without a boundary. So this is basically cancer. If we look at a normal cell and cancer cell, you can clearly see that the difference between these two cells in their appearance, in their morphology. And now what happened? When we were born, this human race came in this world by evolutionary biology millions of years before. Then Mother Nature took care of these natural defense systems to protect us from many these different sorts of these antigens, pathogens, which are available. And then that means the body defense systems is a result of this evolutionary biology system. Now the question is, if cancer is so deadly, and if cancer can cause the death of human species, then why did evolution did not take care of these defective genes millions of years ago by natural selection? Usually this is a joke by Mother Nature. Most of the cases, cancer appears, the probability of appearing cancer, this maximum for cases when our reproductory system is, a function of our reproductive system is over. That means it is ensured that we are, this creating our offspring, offshoots, and then the body can be affected by cancers. So there is a evolutionary pressure to, so there is less evolutionary pressure. So evolution or evolutionary biology doesn't take care of this cancer because already this race is continuing. Human races, human species is there. Further new species has been born, and then because the reproductive system is over, then the chances of getting cancer is more. So the cancer evolution, basically, if you look at this cancer evolution, so there may be different pathway. After this irregularity starts, that there can be different, different pathway. Niche 1, niche 2, Sometimes it is a case of relapsing in microenvironment. Sometimes it is called metastasis. And metastasis, basically, it is after it forms at certain place, it spreads from original sites to other parts of the body. It breaks from the cancer cells breaks from primary tumor and then travel through the bloodstream or lymphatic systems and from new tumors and some other organs or tissues. These new tumors are made of cancer cells, and these are similar to primary cancer. For example, metastatic breast cancer in the lung, it can happen, but still it is breast cancer. And then this is basically this lung cancer. If it is a cancer at lung, it can spread to adrenal gland, it can spread to bone, brain, liver, other lungs. Still it is lung cancer. If it is colorectal cancer, it can spread to liver or lungs. Still it is colorectal cancer. So that means when this metastatic tumor, these are the same type of original cancer is found in other parts. For this is referred to as stage 4 cancer. And many metastatic cancer cannot be cured. So, but with the treatment, their growth rate can be suppressed with some relief symptoms, and life can be extended to some extent. Now, here quantum computer has a big role, or the crucial role to play. Now, evolution progress through natural selection and chance, because this is nothing but molecular mechanisms. These molecular mechanisms that makes the life possible are the product of random mutations over billions of years of trial and error. Now, we cannot expect our body to mount a perfect defense against this deadly disease because large number of mutations involved. So quantum computer can process huge, this gargantuan information. This is not possible with these digital computers or even supercomputer. Quantum computer can process tremendous amounts of information, vast amount of information, and identify what is the root cause of it. So it is ideally suited to attack a disease because we are confused. How did it happen? What is the origin? Then everything, if it can be processed through quantum computer, then we can understand the entire mechanisms for the cancer from the root itself, and there is a chance of this nipping in the bud. And now quantum computer is showing us new insights for treatment of incurable disease like, besides cancer, Alzheimer's disease, where it is related to our this brain, Parkinson disease. In Alzheimer's disease, you can see this this entire brain morphology changes, Parkinson disease, and this ALS, and many types. All these types of disease. This ALS disease is basically a typical type of disease. This ALS disease, this amyotrophic lateral sclerosis or Lou Gehrig's diseases. Stephen Hawking, he was affected by this disease. It is a progressive neurodegenerative disease, and it affects our motor neurons and on the brain and our spinal cord. That means it causes muscle weakness and eventually paralysis. But the brain functions. This brain cannot control the voluntary muscles. That walking become difficult, talking become difficult, swallowing become difficult, breathing become difficult. But mental functional sense and sight, like sight, they're seeing and hearing, these remain intact. That happened with Hawkins. We have seen that till his last date, he was a prolific researcher. So this is basically this cancer. So liquid biopsies now another technique which I'd like to discuss right now. This is a new things, but before that but many times we do not know we have cancer because the sign of cancer sometimes become ambiguous or hard to detect. By the time a tumor is formed, there may be billions of cancer cells growing in the body. And in a malignant tumor, if it is formed almost, then immediately surgery, radiation, or chemotherapy is recommended. Sometimes it is too late. So there are traditional treatments like surgery, bone marrow transplant, chemotherapy, immunotherapy, radiation therapy. All these are, but these latest things is immunotherapy that we will discuss later. Now, simple blood test. We use most of the cases to identify whether we have increased sugar level or diabetes or all these things, but we cannot detect cancer by the pathogenic test, simple pathogenic. Why it is so? Because our immune system cannot detect cancer cell. They are our own body part. So they are not like foreign body or antigen. So that's why they evade this radar system. Bodies, these immunologic radar systems. So it is not recognized by immune systems. These are own cell and they have gone bad and they elude detection. Therefore, normal blood test cannot detect cancer. Now we have immune systems at many places: mucous membrane, lymphatic vessel, thymus, skin, bone marrow, spleen. There are many areas we have our immune systems, but still these immune systems cannot detect, this cancer. Now we know that for the last 100 years, these cancer tumors, they shed cells, and these cells, the molecules, and these cells and molecules, they are observed, they are present in our body fluid like blood, urine, cerebrospinal fluid, saliva. But this detection is possible when there are billions of cancer cells growing in our body, so the population is, then that time we can detect. By this time, we do not have any other option except surgery to remove the tumors. Now, genetic engineering, through genetic engineering, we can detect the cancer cell protein in our blood streams or body fluid in low concentration also. This that means if initial detection is possible, then subsequent the tumor formation, all these things can be prevented, and this is called liquid biopsy. This is a very fast, convenient, and versatile way of detecting cancer. It is based on this genetic engineering, and this in recent year, the clinical development of liquid biopsies for cancer, it is a revolutionary screening tool that has created great optimisms. It is reported by Lisco and Jenna Aronson, American Journal of Managed Care. And now with liquid biopsies, we can detect over 50 different types of cancer. So this basically it is related to starting of DNA, starting of RNA, DNA abnormality, RNA expression, protein expression, amplification, then translocations. All these things are detected in liquid biopsies, and then we can study this genetic pattern, and then I can identify whether cancer cell has already been formed. And in future, commercially, we'll be seeing toilet in our bathroom. It would be sensitive enough to detect signs of cancer cells, enzymes, and genes circulating in bodily fluid. When every time we go to the bathroom, we might be unwittingly tested for cancer. Already, this AI toilet has already been developed. It takes photos of our poop to check our health. And in Japan, they have developed smart toilet systems that analyze your waste and bowel movements, that sends this personalized health reports and lifestyle recommendations to us again through mobile phone. All the data is transferred to cloud, and the cloud, this genetic, it is genetic through the genetic mapping and matching, this cancer identification takes place. Now this requires, you see, we are now 140 billion people. And every time we go to toilet and our, this all these things analyzed and taken care of, that means real-time basis, it is huge amount of data. And 140 people's genome that is to be kept as a library or record. So every time our body fluid is tested, that entire the genetic things and is mapped with this particular genomic pattern, and then we can identify this. There is abnormality, and at that level cancer has happened. But it's a huge amount of data. This huge amount of data can be handled only by quantum computer. So quantum computer is, this will be useful in detecting cancer at the very, very early stage without our, without, we were not aware that this has happened. But normally, immediately it will be detected, immediately all this treatment will be started, and immediately will be relieved of it. So that means our genome daily basis or weekly basis is scanned by distant quantum computer to find an evidence of harmful mutation. This will not be a cure, but it will allow to prevent it from spreading. because the cancer problem is with detection takes place at the last stage only. The spreading process has been, this done in a significant way. That's why afterwards cancer will be, this a common nuisance, harmless nuisance like common flu. Now this there are many advantages of liquid biopsies. It is non-invasive. It is less expensive than tumor biopsy. It detects material straight from multiple tumor sites. It can be obtained serially for screening, determining therapeutic response and assessing for minimal residual disease. And then sometimes low amount of tumor DNA set, if it is there, detection sometimes is difficult at very low level of concentrations. Reproducibility sometimes is an issue. So there are many cases when we grow up, we become old, that our, this basically our own body cells, they, this, that is not cancer, but it discharges certain cells which is aged cells and which may have certain resemblance with the cancer cell, but this is not cancer. Sometimes there is, that's why there is some falsification. So these are the initial problem, but it is becoming more and more mature technology, and within few years, this liquid biopsy will be this readily available technology at different pathological labs. But the thing is this analysis of the data, and for that quantum computer is required. Now I compared at near future, it will be there's not so harmful nuisance like common cold. But can we cure common cold? Actually, we can cure, but there are over 300 rhinovirus that cause cold, and they constantly mutate. Now that means we need to take 300 vaccines. Therefore, this preventing ourselves from the guarding ourselves from this common cold. That is a cumbersome effort. So that's why we simply live with it. We have learned how to live with common cold. Now in future, out of all these things, liquid biopsies, analysis of data and all these things, and then detections, early detections through quantum computing and all these things, so being a death sentence, it may eventually be viewed as a nuisance. Now there are so many cancer genes. Now it might be impractical to devise cures for all of them. But if we can detect them long before they spread, it might be possible to stop their progression. So that means we can have cancer, but we will not be dead. So this is the future scenario for the cancer. Now, if we look at quantum computing, why it can handle so much humongous quantity of data? Because classical computing, those who have certain knowledge of computer science, it works on these principles of bit 0, 1, off on this binary mode. But in quantum computing, it's not like that. It is basically this scenario of qubit for quantum computing. This entire this volume. Here the possibility of 0 and where everywhere. That's why we now, instead of bit, we characterize or designate quantum computer with qubit. And last report which I am having, there are many the computer commercially available computer that may come in market. IBM, I told that IBM is leading the race, and their quantum computer Osprey. Osprey is their quantum computer that is coming in the market. Then China, Zhuheng, China is also on the race. Then Google is there. Bristlecone, that is their quantum computer, name of quantum computer. Google has two quantum computers. One is Sycamore, one is Bristlecone. And then Intel has Tangle Lake. So initial report, even in 2021-22, the report which I had, that IBM Osprey, it is now dealing with 433 qubit. That was the highest. And Intel Tangle Lake, that had the lowest qubit, 49 qubit. But recently, there is a report that IBM already, this that is that one then bringing in the market one complete quantum Condor, and that has more than 1,000, 1121 qubit. So this has a huge processing powers. So this is quantum computers, how these are functioning, what are the different mechanisms for this designing this quantum computer. There are 10 or 15 different ways of making this quantum computer, and many countries are working on different mechanisms. Ultimately, it is the coherence of these waves. That may be this optical wave, that may be electronic wave, that may be the sonic wave, any waves. But that is a different topic. So advantage of quantum computer is its speed and efficiency. It can solve any complex problem, anything, that's why we are scared about our this cryptography and our data security. But the most important thing, which as a this boon for human race, it is bringing, that is advancement in medicines and health. There are other aspects also, the supply chain and all these things. You can use quantum computer to improve stronger cyber security. That there will be another race. So how to make it more strong, how to make it more vulnerable. And then the climate change and sustainability, it is another area where quantum computer can be used to understand the interpattern. That these are these big areas. Now the thing is, there is another way how to detect cancer. That is by sniffing. Sniffing means if we have cancerous cells, our body odor will change. If we have any disease, our body odor will change. And that change can be detected by sniffing, by our olfactory systems, by mechanisms. Now in future, it is being said that cell phone with odor sensor, it can be connected with quantum computer in cloud that can help in detecting cancer. That is, there's another step ahead from liquid biopsies. This mobile phone will be in our hand all the time. We are inhaling and exhaling breaths. It can detect the breath and immediately it can transfer the data. That means at the very at the incipient stage, at the this stage of onset of this cancerous cell formations, these vibrations, the information will be passed on to the cloud. This now we see during COVID period, probably you are aware, in Germany airports, dogs were used to detect coronavirus, because normal PCR test that requires few days, and dogs take few seconds to detect whether any person is traveling with this active coronavirus. And dogs can be trained to identify lung cancer, breast cancer, ovarian cancer, bladder cancer, prostate cancers. They're 99%, 98% accuracy. Sometimes our traditional, this analysis technique do not have that sort of accuracy. Why it is so? Because dogs are 220 million nasal scent receptors. We have only 5 millions, 40 times more than us. That is not the only fact. Another factor is they are part of the brain where all the data is processed is much larger than our brain. So that's why they can, this unleash the true potential of their olfactory power. But what is the problem with the dog then? The problem is we need to take few months to train the dog, and there is a limited supply of these varieties of dogs who can be trained properly. So that is one of the problem. It was thought that we would be training dogs for treating cancer, for identifying cancers. But afterwards, there is an organization. This organization is called DARPA, Defense Advanced Research Project Agency. They basically formulate all the problems of NASA. They sponsored a contest for creating robotic nose or nano nose. There is this nanotechnology is going to be another this game-changing technology which is coming in near future. A brilliant scientist of MIT, Andy Marshall, he first tried. He was this amazed to see the power of these dogs. Then he imagined these applications of nano nose or nano sensors in that form, which can detect this odor and other elements, and which can be connected with cell phones for giving alerts. And there's a collaborative research work between MIT and Johns Hopkins University, and ultimately they developed this sensors. And this is 200 times more sensitive than a dog's nose. That means with 100% accuracy, it can detect the cancer, but it is still costly. It is last information tells that it cost till now $1,000 to analyze one sample. It is expensive for a country like us, but maybe in the future, because the technology has already been developed, it is a matter of time. It becomes cheap. It becomes cheaper and cheaper, and accessible to billions of people across the world. Now, immunotherapy, this is the latest techniques for treating cancer. Main treatment of cancer is right now surgery, where tumor is cut, radiation. Here, X-ray particularly or particle beam is used to kill the cancer cell, and chemotherapy to poison the cancer cell. I told at the beginning of my lecture, this is not a total curing process, because they are attacking a zone where the probability of this cancer cell is the maximum, but that doesn't mean the probability is not is absent elsewhere. So when we are killing some major cells at some places, at other parts, already cancer cells are migrated and they are forming there, or some cells are still remaining there and they are relapsing. So that's why many cases we see that even after surgery or radiation or chemotherapy, it relapses, or it transforms in it undergoes this different transformations. But immunotherapy, this is used for cancer of the bladder, brain, breast, cervix, colon, rectum, esophagus, kidney, liver, lung, lymph, skin, ovary, pancreas, prostate, bone, stomach, and leukemia. There are different degrees of success. Now, this immunotherapy means here cancer cell cannot easily be identified by the body's immune system, because I told earlier, it's a part of our body system. Body has two types of cells, T cells and B cells. These are programmed to identify and kill foreign antigens. B cells, they attack invader outside the cells, and T cells, they attack infected cells. But they cannot detect, because the cancer, as they are part of our body, they are not foreign body, so they escape their scanning process. Now, in immunotherapy trick, it is to artificially boost the power of our immune system to detect this cell. Now, here what the mechanism is: genome of the cancer is first sequenced, and precisely types of cancer being started and how it is developing is known. Then WBC, or white blood corpuscles, are extracted from our blood. Genes from the cancers are processed. Genetic informations of the cancer is inserted into white blood cells by virus. Virus is which is being rendered harmless earlier. So it is a carrier. It can carry the information inside the WBC. So that means WBC are now reprogrammed to identify cancer cells. Now this WBC again injected back to our body. This is the mechanisms. That means, this entire cancer, first analyzing the entire, this cancer pattern, these genomic patterns, and then inserting this pattern under WBC. This is available only, but there are limitations. Limitation is available for some cancers. But I told earlier, cancers are so many types. So now there is, the encyclopedia for different varieties of cancers. Now, sometimes these WBCs, as they are modified artificially, the modification is not perfect, and sometimes this may cause a certain, the side effect, and sometimes it will be fatal. And there are paradox of our immune system, because essentially there are unlimited numbers of viruses and bacteria. But how our immune system tells the difference between dangerous and friendly one? Some bacteria, they are staying inside our body, in our guts and other places. They're benign. So how we are allowing them to stay, and how we are fighting, our body system fighting with other bacteria, other viruses. This is like a police that knows whom to arrest and whom not. This evolution, it is basically taking care of this problem. This B white cell, this contains Y-shaped antigen. You can see from the diagram, Y-shaped antigen receptor. This protrudes from the cell walls. And WBC latches the tips of the Y receptor to the dangerous antigen for destroying it. When WBC is bound with the genetic code in the tips of the Y receptor that matches the receptors of the specific antigens, these are randomly mixed. In principle, basically all the codes that the body may ever encounter are already contained in the various random Y receptors, those which are good, those which are bad. And the paradox is, let us we have small number of amino acids that can create a large number of genetic code. How this is so? Let us take, we have 20 different, there are 20 different amino acids in human body. To create a chain of 10 amino acids with 20 possible amino acids in each slot. So there will be 20 to the power 10 possible random arrangement of amino acids. Now, actual number of B cell receptors has about 10 to the power 12 different possible combination. Which one is higher? Between 20 to the power 10, 10 to the power 12. If you look at it, it is basically 2 to the power 10 into 10 to the power 10. 2 to the power 10 means 1028. 1028 into 10 to the power 10. And it is basically 10 to the power 2 into 10 to the power 10. That is 100 into 10 to the power 10. That means it is 10 times more. So covered the entire things, entire gamut. This astronomical number contains all the possible antigens it may encounter into the. So once Y receptors are all randomized that contains the genetic code of the body's own amino acids, they are removed. They are allowed to enter. Left behind the Y receptor that only contain the genetic codes of the dangerous antigens. So this is the very important part, because these Y receptors first identify entire things which these our bodies associated with the body, allow it to pass, and then those Y receptors which do not detect this body's code and all these things, that they are left behind to tackle the dangerous antigens. So that means it is like a, this policing activity. Police, when it is finding a huge crowd of people, first they identify previously known innocent people. Then they know that the criminals might be among the leftover people. Now this paradox is, sometimes when deleting the genetic codes found in the body does not remove them all. It happens in police world also. Sometimes good codes are left behind, like these some innocent people are this left behind with the criminals. That means the police then attack these innocent people also. This is the reason for our autoimmune disease, like rheumatoid arthritis, lupus, type 1 diabetes, multiple sclerosis. These are all autoimmune disease. Here, some good genetic codes have not been identified by the system. Sometimes the reverse happens. That means, by mistake, the immune system not only removes the good codes, it removes some bad codes. Then the immune system cannot identify the dangerous antigen. This happens in case of certain cancers, when the body is unable to detect the antigens with the wrong genes. This is one probability. Now, entire process of identifying dangerous antigens is purely quantum mechanical. The digital quantum computers they cannot, they are not capable of reproducing complex sequence of events at the molecular level for the immune systems to work properly. These huge numbers of steps are involved. Quantum computer, they are powerful enough to unravel molecule for molecule the function of immune system. So this is the one of the biggest advantage. And of the latest technique, CRISPR, probably you have already heard this CRISPR, this therapeutic application of quantum computer. This may be increased when it is combined with CRISPR. CRISPR is an acronym of clusters regularly interspersed short palindromic repeats. This allows scientists to cut and paste genes. And quantum computers, these will be able to identify and isolate complex genetic disorder. Then CRISPR then used to, that means for identification by quantum computer, then using CRISPR to cure them. So CRISPR, they have this works by using a guide RNA, gRNA, and to direct the Cas9 enzyme. This acts like a molecular scissor, and to a specific DNA sequence. Once there, Cas9 makes a double strand cut in the DNA, and this then cells natural repair mechanisms, machinery that takes place, and which can be used to disable the gene or to insert, delete, or modify genetic materials. So first is recognition, and then recognition by quantum computer, then designing of this gRNA or guide RNA that is complementary to a specific targeted DNA sequence. Then attachment of this gRNA with Cas9 enzyme with formation of complex. Then cleavage of this at the correct spot. It is similar to this lock and key work. When Cas9 finds a target, it creates a double strand break and then it repairs. So this is basically the three components are involved. First, quantum computer, then CRISPR, which involves Cas9 and guide RNA. So this research, particularly now, it is very much promising in treating cancer of future. And earlier research, it was not this target specifics, and that's why earlier research was this broad area research, but it is a focused and targeted and specific this focused research and focused area where this cancer will be treated, and not only cancer. Besides this sickle cell anemia, cystic fibrosis, Tay-Sachs, these are the diseases at many places of the world, Africa, Northern Europe, geospatials. And there, this genetic treatment and quantum computer combinations, now it is giving the new ray of hope. Now to a lighter mode. Genetic disease is very, very, this important in human race, because in many, we have seen that genetic disorder, it changed the course of history. These royal families, they used to engage themselves in basically intermarriage. Because of intermarrying, these genetic diseases are prevalent in royal families of Europe. At one point in time, US was under England. And that time, King George III of England, he suffered from a genetic disease that rendered him mad. As he became mad, so he could not take care of the grievances of people of America, and that led to American Revolution with the leadership of George Washington, and that rest is big history. America became America, out of the clutch of this Britain. Similar thing happened at this Russia. If we look at this Russian history, the son of Tsar Nicholas II of Russia, he was afflicted with hemophilia. And then there was no treatment. So royal family, they believe that can be treated by a mystic. That mystic is Rasputin. Rasputin, it's a corner for Rasputin. This Rasputin, he was a cunning and notorious persons, and he engaged in the royal family in all other these luxurious activities, and the royal family, this Tsar, they became detached from the common masses. The monarchy were paralyzed, and reforms was not done at proper time, and that this resulted in Russian Revolution 1917, where Lenin through Bolshevik Party took the power and established the communist regime in Russia. So that is the history. So there are peculiar things, sometimes genetic patterns, sometimes environment, that affected the course of this history. Now in immunotherapy, first the desired gene is inserted in harmless virus, then the virus injected the patient then infected desired gene. But I told earlier, the body sometimes would often recognize the virus, attack it, causing unwanted side effect for the patient. In 1999, before that, there was a big hype regarding immunotherapy. But when this gene therapy, when a patient died after a trial in 1999, there was this busting of the bubbles, and basically immunotherapists, the researcher immunotherapists, they thought that this is not the right way. Then this CRISPR technology developed in 2018-19 only. And that is a more precise. That is the more precise one. This you can see these two ladies. One is Emmanuel Charpentier and Jennifer Doudna. Left side is Jennifer Doudna. There is a famous book, Code Breaker, by Walter Isaacson. You can go through that book. How they had done their research works in this developing this CRISPR-Cas9 technology. But the cue came from Mother Nature, because virus attacks bacteria also. When virus attacks bacteria counterattacks virus, releasing a barrage of chemicals that split the genes of the virus at precise point. stopping infection. This mechanism was isolated and this used to sever viral genetic codes and at desired points. So they got the Nobel Prize for this, for CRISPR technology. And it opened a floodgate for genetic engineering. Now quantum computer is coming. So this combination will be, this very much, formidable in treating cancers and all sorts of diseases like this. Now we have a big gene. This is P53, which is called guardian of the genome. P53 basically it is a tumor suppressor gene. It is required, it is present in everyone's body. It is vital for stopping the growth of cancer. But it's a big gene. And as because it is big gene, so there are many sites available here where mutation can take place. And because of mutation, it is also responsible for half of the common cancer, like breast cancer, colon cancer, liver cancer, lung cancer, ovaries cancer. So that is basically related to mutation and specific sites. For example, long-time smokers. Long-time smoking often develop cancers at 3 specific mutations along P53. That means it proves that person's lung cancer most likely to come from this cigarette smoking. So this P53, when it is a long gene, it is if we would like to cut this gene using immunotherapy by broad-based technology, it will be, most of it will not give the desired result. But quantum computer can potentially decipher and map molecules within the genes of the cancer cells. Then CRISPR can be used to this target at that at the specific point to cut the genes at precise points. So gene therapy, quantum computer CRISPRs, this make it possible to cut and splice genes with ultimate precision, reducing the problems of lethal side effects. Now, CRISPR gene theory, now there is a very famous quote by Clara Rodriguez Fernandez. She is a very famous science popularizing article writer. In Lamp Biotics, he commented, in theory, CRISPR could let us edit any genetic mutation at will to cure any disease with a genetic origin, with over 10,000 diseases caused by mutation in a single human gene. CRISPR offers hope to cure all of them by repairing any genetic errors behind them. You can understand this involves processing of huge number of data. And that processing is only possible to the best possible extent by quantum computer alone. Now there are so many success stories because it is a recent phenomenon. CRISPR came only a few years before. At University of Pennsylvania, scientists used CRISPR technique to remove 3 genes that allow cancer cells to evade body's immune systems. Then they added another gene that can help the immune system to recognize tumor. This was made as a safe methods, and even on patients with advanced stage of cancer, this method was applied with success. Now it is running a test on number of blood cancer patients, this CRISPR therapeutics. These patients are being treated with immunotherapy, which uses CRISPR to modify their DNA. Now, sickle cell anemia, CRISPR therapeutics is harvesting bone marrow stem cells from patients suffering from sickle cell anemia. CRISPR is altering these cells to produce fetal hemoglobin. These treated cells are then inserted back into the body. So this is one of the areas. Sickle cell anemia was a non-curable disease even a few years before. AIDS. A small number, now you see this for the AIDS, a small number of individuals, they are born with natural immunity to AIDS because of a mutation in their CCR5 genes. CCR5 genes actually is responsible this for AIDS because the proteins made by these genes, it creates an entry points for the AIDS virus to enter a cell. Now in these rare individuals where there is a mutated CCR5, that gene can be identified and that gene can be used to insert in other people's body, other person's body to give them immunity against the AIDS. So this is one of the treatment for this AIDS. Cystic fibrosis. Cystic fibrosis is a relatively common respiratory disease, and it happened in our lung systems. They suffer, and they rarely live beyond 40 years. Because those who are suffering from cystic fibrosis. It is a mutation in CFTR gene. Now, doctors are using this CRISPR to in Netherlands, their success story to repair this gene without the side effect. Already now there is a commercial firms, Editas Medicare, CRISPR Therapeutics, Beam Therapeutics, they are treating this cystic fibrosis with CRISPR. Another disease is Huntington's disease. This disease, the reference is found in history also. It is associated with dementia, mental illness, impaired cognitions, and other debilitating symptoms. There's a story of Salem of US, that there are some women, ladies were there. They were under trials, suffering from disease and with tortures and all these things. And it was that time, the idea was, the common notion was, these ladies, they were behaving abnormally because they were bewitched. So in the name of this removing this ghost from these ladies, they were exposed to tortures and all these things. But actually they were suffering from Huntington's disease, and that created certain the cognition problems and dementia. That's why they behaved abnormally. Here also, this many times we have seen, we heard the cases of Huntington because with this abnormality in cognitions and dementia. So this is not something abnormal or this something related to spirit and ghost. It is a genetic problem. And in Philadelphia, at Children's Hospital, scientists, they use CRISPR to treat this disease. Now, there are certain problems. For example, CRISPR can create, can be, this good treatment process for all these diseases which I have mentioned. But there are certain diseases, schizophrenia, they involve large numbers of mutation, plus interaction with the environment. Now, this requires that proper understanding how this mutation can create an illness at the molecular level. That necessitates the full power of quantum computer. Sundar Pichai now with the Google computer. So once we know the molecular mechanism by which certain protein cause genetic disease, then we can modify it or find more effective treatment. So this can aid and can this further enhance the potential this treatment benefit of CRISPR. Now let's deal with one paradox, Peto's paradox. So far we have discussed that cancer is a result of mutations, and it's this common conclusion that numbers of cells is more, numbers of mutation is more. If numbers of mutation is more, probability of cancer is more. And you see, we have discussed that P53 genes, we have one P53 gene. So, and at one P53 genes is responsible for large numbers of cancer development because this is a long gene. In many places there can be mutation. But biologist Richard Peto of Oxford, he noticed something odd about elephants. Elephants big animal, this animal. So it is quite expected they would have more cancers because of the large size, large numbers of cells, large numbers of cell division, large probability of mutation. But elephant, actually, elephant have a low rate of cancer. So the rate of cancer does not depend on the number of cells. Elephants have 20 copies of P53. That means probability of cancer should be more. Human has 1 copy. Extra 19 copies of P53 of the elephant, it was observed that worked with another gene LIF, and this gives us protections against cancer. So the combination of genes like P53 and LIF, these are thought to suppress cancer in large animals. In the elephant like big animals. But still there is another paradox. What about whales? Blue whales is the largest animal. They have only 1 copy of P53 and 1 version of LIF. They have also very low rate of cancer. That means some unidentified phantom genes are present in these animals, which we have not yet detected, not yet invented. That can be one of the clue how to prevent cancers, because that combinations of that gene along with these genes, it resulted in the development of robust defense mechanisms in these animals against cancer. So in animal kingdom, there could be numerous genes which are protecting against cancers and other diseases, and which this enhancing the lifespan also. Certain sharks, Greenland sharks, they live up to 500 years. Certain tortoises, they live up to 400, 500 years. So this is related to their genes. So if we can identify those genes and these genes can be inserted in human bodies, then we can get these sorts of benefits which these animals are already having by from Mother Nature. So the hope is that by seeing how evolution has found a way to prevent cancer, we could translate that into better cancer prevention. Every organism that evolved large body size has a different solution to Peto's paradox. There's a bunch of discoveries that are just waiting for us out there in nature, where nature is showing us the way to prevent cancer. That is the comment of Carlo Mallet, who is researcher of P53 genes of animal kingdom. So quantum computer may prove instrumental in finding these mysterious anti-cancer genes. So there are many ways in which quantum computer can work. Liquid biopsies, already I told, the processing of huge amount of data, that it can detect cancer cells years before tumors form. It can make possible a gigantic national repository of up to the minute genomic data using our bathroom to scan the entire population for the earliest signs of cancer cell. If cancer from quantum computer enable modification to our immune system, that would allow it to attack hundreds of different types of cancer. And the combination of gene therapy, immunotherapy, quantum computer, CRISPR, these are potentially cut and paste cancer genes with great molecular precision that can help to reduce the lethal side effects of immunotherapy. So genes like P53 are involved in first major cancer. So gene therapy combined with the new insights that can, this from quantum computer may be able to stop it. Now, seeing all the prospect of quantum computer, of CRISPRs and genetic engineering and all these things, President Biden, in 2022, he declared a cancer moonshot project, a national, this Nixon announced in 1971, based on the limited success of traditional cancer research. But seeing the potential of these advanced technologies, President Biden he declared a cancer moonshot, this national goal to reduce the cancer death rate by at least 50% over the next 25 years. And given the rapid advances to biotechnology, it is a formidable, achievable goals. Then what will happen to cancer? We will suffer from cancer because there are many ways to form cancer. But in future, this cancer, as told earlier, it will be treated like a common cold as a preventable nuisance. Now another thing is coming to make it more precise. That is a combination of artificial intelligence. Artificial intelligence and quantum computing combination that can help us in creating designer proteins out of which our bodies are made. And it will give us a line of defense against this disease. So this AI and quantum computing that will enable us to cure incurable disease and reshape our life itself. So this is the end of presentation. So there's these tremendous potentials in future of these upcoming technologies, quantum computer, CRISPRs, and artificial intelligence, and all these things. So I tried to make the present one glimpse of all these things in a nutshell. So now this is the end of it.
Thank you, sir. It was really an enlightening session where we came to know about the historical perspective of cancer, the emerging field of quantum mechanics, quantum computers, artificial intelligence, and smart toilets, and the use of odoriferous molecules for sniffing cancers, the robotic nose. Then we came to know about immunotherapy and the CRISPR technology and the Peto's paradox. And last but not the least, we also were enlightened about the cancer moonshot, which was launched by President Biden. Now, I would request the participants, if they have any questions, they may raise their hands or they may post their queries in the chat box, and they will be taken one after another. Thank you. Oh, please move ahead and ask questions if you have any. I request the participants.
or you can send question to me by
Myself,
email.
Dr. Deblina Sharkar from Bagnan College, sir.
Okay, okay, okay.
This is not related to research. Actually, dogs, they suffer a lot from colorectal rectal cancer.
Yes, yes, yes.
Does this have a definite reason that why do
Yeah,
they?
that you see the difference between our colon and length of our colon and dog's colon. Dogs or any carnivorous animals' colon is that because you see, their colon length is small. Our length is big. Actually, we are not designed to eat meats and all these things. They were designed because this basically when we take meats and all these animal products, the formation of amines is more. Amine, this at the molecular level, amine is more responsible for creating cancer. Amine is nitrogenous. Well, nitrogenous is the adsorptions and then cell mutations and all these things. Now, why our poops is so this obnoxious? Because of this presence of amines, different sorts of amines. Now their system is small and they immediately can discharge the entire thing from their body. Our system is difficult. We used to say the European suffers from this colon cancer because of their food habit. They used to take lots of meats and all these things. Of the late, colorectal cancer in dogs has increased because of the this food scarcity, and sometimes they are, because it is more prevalent in stray dogs, they are taking foods from dustbins and trash, these trash cans. There the food is contaminated with plastic, processed foods and all these things. So that means these amines and all these things, it is again getting transformed into this a different form, different molecular form. So that's why it is all these things can be afterwards at everything is happening at molecular level. So my observation is that this appearance of cancer in stray dogs of the late in India has increased because of the food habit of ours. We are discharging processed foods, we are discharging all these things, and we are discharging, we are not also taking care of this segregation of waste. Dogs are eating all these things, plastics are entering in the body. So we need to study all these things. Why there are chances? Because of the late I have come across certain cases like this of the stray dogs, the cancer. Earlier it was not so, but I am associated with an NGO. They are telling that they are getting more cases of the dog cancer because of their these foods, and this ultimately responsibility is ours. We are feeding them with such a this contagious and the contaminated food with sorts of this processed foods and these plastics and all these things. So this is happening to them. But the mechanism, what is the exact mechanism, that needs to be studied, and for that this maybe this that requires a huge data analysis, and that is possible this maybe when the quantum computer will come in picture, then we can will be in a position to know at molecular level what is happening actually.
Okay, sir. Thank you.
So that is, as I told earlier, cancer is not a this one factor is only responsible. There are multi factors. Pollution is one of the factor of the late. This polluted air, polluted food, polluted water. This all these things are responsible. But exact what is the mechanism which is happening at the molecular level, that we need to study.
Yes, sir. Actually, rectal and colorectal cancer rates, they are also increasing in youth.
Yes, yes, yes. That is increasing. That is increasing because of these foods. In this not only with dogs, even if we look at this human parts also. Our this talks because of this problem with our food, a problem with our adult, this environment problems. So there are many factors broadly identified, but at molecular level, how these factors are combining to develop the result outcome, that mechanism is still to be studied. There are many hypotheses, but these are all broad based. We know that these are the different pollutants, these are the the lifestyle habits, these habits, that. But what is actually happening as for the development of these cancer cells? That cancer basically, you see, if everything in coherence, then there is no possibility of cancer. Coherence, that is the basic mechanism of quantum mechanics. Quantum mechanics is coherence, associated coherence. If the vibration, this coherence of the vibration matches, then this is this is a quantum phenomena. And if it is decoherent, then it is not observable. So sometimes this vibration is not matching and abnormality is developing. So that is at the vibration level. Afterwards, this we need to understand many things.
Sir, there is a question from Miss Sujata Dhar Dotto. She asks, is there any type of cancer that cannot be treated by immunotherapy?
There are many types of cancer. Immunotherapy has a limited success only. Right now, what is happening, if you look at if you study the literature, cancers, these thousands and thousands, new and new forms of cancers are being developed. So many types of cancers are coming up without any prior treatment right now. Now that's why we are, this presuming, or we are foreseeing the potential of quantum computer. If we can have the entire data, if we have the processing of the data, and if we can identify the mechanisms which is happening at the molecular level, then it will be easier for using, this immunotherapy or CRISPR technology to specifically target that thing. Now, there are lots of things we need to study now in mother nature, in animal kingdom, in plant. There are many genes available. We do not know the exact nature of the genes, which genes is working in which way. I told you that in case of elephant, one gene is working with p53. In case of whale, this we do not know what type of gene is working with p53. A combined effect. So these phantom genes or unidentified genes needs to be identified first. So this is the huge work. After that, we can know the combination of which genes, which insertion of which genes, deletion of which genes is required to make the treatment more specific.
Okay. Sir, we have another question from Chandan Singh. He asks, after long research and funding, still conclusive treatment option is not there. How that is going to affect future funding?
Actually, you see, this is the problem with, this, I saw you, the moonshot project of President Biden. But the present president of US, he is not believer of scientific research. So far, I came to know, or probably you have also this information. Even NASA fund, or this allotment that has been reduced. NASA's funding for research projects and all these things, university they are also. So, and he never believes that environment can have a deleterious effect, and the global warming is a problem. He doesn't believe in this concept. That's why we need to have people at the helm of affairs with proper scientific understanding. Technology is there, but to push that technology to make it this available commercial, because you see in any technological scenario, initially it is costly, but after that it becomes more and more cheap. Now, for that, it needs a push, and this push that can happen only at research labs, only sufficient, adequate amount of funding. It's a sort of state of affairs that the people who are supposed to look after these fundings and allotment and all these things, they are not appreciative of this research, importance of research and all these things. But let's hope for the best. We know these corporates, because IBM, Googles, and others, they are working in a big way. They have their, these many IT companies, they have their biological wings also. They are working on the computer, this quantum computer, along with the gene research together, and development of these designer medicines, vaccines, and all these things. These are coming up. Initially, may not be affordable for a country like India. As I told that, the sniffing of this cancer, it costs $1,000. $1,000 means in Indian currency, it is almost 1 lakh. So one test costs 1 lakh. So how many people can afford that? So that's why this initially at this moment it is not affordable, but it happened with other technologies also. Initially not affordable, even this desktop and all these things which we are using right now. Earlier it was not so this affordable. Right now this smartphone, desktop, these are becoming more and more affordable. Similarly, we hope that in future the push of the power of technology will make it affordable and accessible to the billions, among the billions of people.
Sir, Dr. Namrata Roy wants to know the CRISPR technique in a bit more detailed way.
CRISPR technique in a more detailed way, you need to study literature. CRISPR technique is this advanced technique. I am suggesting you to read a book that is a very interesting book. You can understand, this the life and work of Jennifer Doudna and Emmanuel Charpentier. Both of them got the Nobel Prize. CRISPR basically it is targeted gene editing. That is, this happening through, this with the protein. That initial, this signal or initial understanding came from defense mechanism of bacteria. So when virus attack bacteria releases chemical to attack the RNA, of these viruses. So similarly, here also, this one in the Cas9 with guided RNA, one guide is produced. This guide and this Cas9, this is a complex, and this is targeted to a specific point of the intergenetic sequence to cut it like a scissors, and then again combining it with by natural mechanisms or by inserting another things. So it is something like molecular level scissoring technique, cutting, pasting, deleting, this joining. So there are plenties of literature available. You can go through it, and very interesting technique. But earlier it was not that. Earlier it was used to. Now it is using RNA this target. Earlier with DNA it was used, and that was broad spectrum sort of thing. And that's why targeted part was not affected. In the double helical strand, the particular site was not affected. It is a the broad-based attack. That is a precision attack with CRISPR. So you can study many literatures. There are many literatures available. You can go through it and understand this CRISPR-Cas9 techniques for gene editing, which is being used. Whereas it is not so complicated, but technically it is the technology is really very, very interesting.
Okay. So we have reached the end of our session. I hope that all the queries of our participants were answered. And I would like to thank Dr. Shoykot Moitro once again for your such an interesting topic. And we would like to invite you again if we have any programs in the future. With that, I would like to wrap up for our lunch break. And I would like to request the participants to join at least 5 minutes before the next session begins, which begins at 2 p.m. And then we can learn something more. Okay, that's all. Thank you.
Okay, I'm leaving then. Thank you very much for your patient sharing. Thank you very much, the organizing team.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir.